No workshop, the
Okay. So, good morning, everybody. Today, we will have further discussions into the concepts that are fundamental for constructing an ecological model. I would like to get back from where we stopped yesterday. As we have seen, or as I already told, all the bases, energetical basis for all the biological processes taking place into the biosphere come from the sun. How do we measure this energy? We started by observing that a measure of solar energy arriving onto the sun may be gathered by employing the solar constant. The solar constant is an indicator, a kind of temperature indicator of the activity on the sun, and measures the density energy flows from the surface, from the sun to the surface of the planet. The value of the solar constant is 1360 watt per square meters. Watt means energy flows. Per square meters means density of energy flows. And if we make 100% with energy, as soon as this energy arrives on the surface of a planet, this energy is differently absorbed or processed by the biosphere. Let's say that of the 100% of the energy arriving on the atmosphere, 35% is immediately reflected by dust, gas clouds, and albedo. Only 46% directly is absorbed directly by the Earth, while 19% is absorbed by the atmosphere. And this quota is what a part of this quota is responsible for the greenhouse effect. It is important to note that the energy flows on the biosphere is in perfect balance. So if we have 100% coming here, 35% is immediately reflected, 19 goes to the atmosphere, 46 goes to the surface of the Earth, generating tides, currents, and so on. And then all this energy is re-emitted so that we have a 65% remission throughout the troposphere, so that 65 plus 35% means 100%. So the system is in balance. All the energy that arrives onto the surface of the planet is re-emitted or reflected by the biosphere itself. It is important to notice that at the level of the biosphere, there are processes of energy transformation. We already said that we will switch our focus from the quantity to the qualities of the energy. In order to understand what we mean by quality of the energy, we may observe that if we measure 1.210 to the 22 kilocalories per year, so all the energy that is directly incident on the surface, so we are converting the solar constant into kilocalories per year. So essentially, this is an integral on the surface and an integral on the complete yearly cycle. So this is the totality of the energy arriving on the surface of a planet each year, measured in kilocalories. Of this energy, terrestrial vegetations and phytoplankton, what we have called primary producers, employ 2.5 10 to the 20 kilocalories and produce or transform this energy in terms of 5.2 10 to the 70 kilocalories year of biomass. So this is the first energy transformation step. What's happening here? Light is being transformed into biomass. So this is really the construction of organic matter, and this is called the primary production. The efficiency of this process is very low. You calculate it simply by making the ratio between the totality of the energy arriving and the energy produced by this machinery, and you end up with a figure of 0.2% efficiency, very low efficiency. We have seen that if we go to the higher trophic levels, that is from the primary production to the herbivores and carnivores, the efficiency grows much, but on the other side, as we will see further later, the quantity decreases. So the point is that while I'm going through the various chains of transformation processes that changes solar energy into organic matter, first primary production and then the upper trophic level herbivores and carnivores, there is a changement of the energy type from solar energy into the energy stored into organic matter. 1 unit of organic matter requires some quantity of solar energy to be produced. The quantity of solar energy for producing 1 unit of organic matter, being it that of primary production, herbivores or carnivore, the request of solar energy increases. So while carnivores biomass is produced, we have a continuous increase of solar energy. And if we make the comparisons on energetical terms, we will see that while the quantity of organic matter produced decreases, the quantity of solar energy needed to produce this organic matter increases. So this is what we mean by different energy types or qualities of energy. And we may say roughly that the highest quality energies are those energies that need more solar energy to be produced by the biosphere. It is important also to understand that the processes of energy transformation, the 2 main processes that you will find ubiquitously or everywhere when dealing with energy transformation at the biosphere, is the first one is production. Production, you may imagine it is photosynthesis. In particular, primary production is photosynthesis directly, the direct transformation of solar energy into biomass. And then to the other hypertrophic levels, we have further synthesis of organic molecules, and the complementary process of production is consumption. Consumption in biology is fermentation or respiration, so oxidation of organic matter. We said that in consumption, during consumption, adenosine triphosphate molecules are produced. These molecules contain energy, so this is the fuel or false cellular processes. ATP is transported all over the cells of the body. We said that we may identify different qualities of organic matter. For example, we may distinguish plants, algae, and invertebrate and vertebrates in terms of their content energy per unit biomass. So we say that plants contain 19.2 kilojoules of energy per gram, while, for example, vertebrates has a larger energy content, up to 26.3 kilojoules per gram. We have also said that we may discuss this distinction also in terms of different types of organic matter. In particular, the main different types of organic molecules are carbohydrates, proteins, and lipids, and it is straightforward to demonstrate or to calculate the fact that lipids have a higher energy content, 37.6 kilojoule per gram, than carbohydrates that have instead the lowest energy content, 16.7 kilojoules per gram. These two set of figures, kilojoule per grams of plants, algae, vertebrate, and kilojoule per grams of carbohydrates, proteins, and lipids, all refers to the same thing by understanding that plants or algae or invertebrates or vertebrates are made of different quantities of carbohydrates, protein, and lipids. In particular, we may notice that vertebrates contain an important quantity of lipids. This is why the energy content of vertebrates is larger than the energy content of plants. Then we switched to a brief, very quick history of the biosphere. And we said that if we look at how evolution has been playing on our planet, we may subdivide the history of evolution into three eras: the prebiotic era, up to from 4.5 to 3.5 billion years ago. So it was the time in which there was no life on the planet. Then we have impressive changement when life appears, and this happened 3.5 billion years ago. And the microcosms of the time in which the life on the biosphere was dominated by microorganisms lasted 2 billion years up to 1.5 billion years ago. And then from 1.5 billion years ago, 1.2 billion years ago, the new era is called the macrocosms. So it is the era in which all the large living forms appeared on the planet. We have said that the bacteria had a relevant role. The bacteria who dominated the planet for 2 billion years, so for at almost 1/3 of its age, have had a dominant role in changing and adjusting the environment in order to foster the development of higher or more complicated living forms. We said that bacteria changed, for example, the atmosphere by regulating the humidity, hydrogen, carbon dioxide concentration, oxygen concentration, and other relevant gases like ozone and so on concentration. All these regulations were made by the bacteria. We made linear projections of these timings on to a 24 hours time interval, so that we realize that while bacteria were dominant for a very large part of this period, let's say the night part, as soon as aerobic respiration was invented, and aerobic respiration is one of the most successful biotechnologies, aerobic respiration machinery dominated for a large part of the daytime. So let's say from 2 billion years ago up to at least 3 billion years ago, so the origin of reptiles, amphibian species, and seeding plants. So this is the era of invertebrate. And since this point, we had a dramatic explosion of evolution process carried carried out by living systems. Now, at this point, we are left with the big questions. What is exactly the effect of this living machinery? What is doing? If we, for example, make a comparative observation of the different atmospheric composition of planets in our solar systems, we have these kind of figures. It is quite impressive to note that, for example, if we look at Venus, the atmosphere, these are composition of atmospheres in the various planets. If we look at Venus, we have, for example, 96.5% CO2 in Venusian atmosphere. If we look at life, instead, we have 0.03% concentration of CO2 in the atmosphere. If we can make some kind of inferences, like, for example, try to see what would happen if on the Earth where there was no life, and our model tells us that in that case the CO2 concentration in the atmosphere would be very, very important, 98.0. So it is evident that life on our living planet is changing dramatically some characteristics of the atmosphere. For example, in terms of concentration of various gases, CO2 or gaseous nitrogen or oxygen, or various other gases. It is also relevant to notice that, for example, the temperature undergoes a very deep process of regulation, so that, for example, on our actual Earth, so on Earth populated by organisms, by an Earth populated by organisms, we have a 14 degree Celsius average temperature. This temperature is the temperature that has been averaged on the whole planet and on the total yearly cycle. So the average temperature on our planet is 14 degrees. If we make the same estimation on a planet with the same characteristics of Earth, but without living organism, our models tells us that the temperature would be much, much higher. Same considerations applies to pressures, to atmospheric pressure. We have a constant 1 bar atmospheric pressure on the Earth, while on the other planets like Venus or the Earth without life, this pressure would be enormous. On the contrary, other planets like Mars, who has a very lower density atmosphere, the pressure would be much lower. So we have seen that living things or living objects are dramatically changing the structure and the constitution of the biosphere. So at this point, it is interesting to stop and to ask ourselves what it is, life. It is a non-trivial question. Still today, we do not have a single answer. I have my idea what is life, and probably I will end up by telling you what is my idea. These are really a personal idea. There is no agreement on how we may define lives. Somebody says that life, for example, goes up to bacteria. Somebody says that viruses that are simpler quantities are even living objects. Of course, from a biological point of view, this depends on which type of processes you are taking into consideration. But this question, what is life, was first addressed in 1944 by a very famous physicist, Erwin Schrödinger. Schrödinger is one of the fathers or of the founders of quantum mechanics. Erwin Schrödinger started by the simple observation that living objects seems to defy the second principle of thermodynamics. What is the second principle of thermodynamics? The second principle of thermodynamics, the so-called law of entropy, may be presented in several flavors, but one way of presenting in a, let's say, in a subtly, in a discussive way is that of saying that the entropy system, sorry, if we introduce some state variables for describing the system, like for example, the entropy is a measure somehow of order or disorder of an object, of a system, and the second law of thermodynamics tells us that spontaneous transformation happens only in the direction of increasing the entropy of the process, of the system, sorry. What does this means? Increasing the entropy means increasing disorder. Entropy is a measure of order, or if you want, complementarily, disorder of a system. Entropy is a measure of the quantity of information that has been embedded or recorded into the structure of your system. In thermodynamics, we know that, for example, if we have two chambers, one empty and the other one totally filled up with a kind of gas, if we connect these chambers with a tube and if we open the connection, then we will see that the gas contained in the first chamber, it starts transferring to the empty chambers, and this process of transfer will end up as soon as the concentration of gases into the two chambers will be exactly equal. So this is a process of transformation that, for example, make a substance, gases, move from one portion of the space-time to another portion of the space-time, and the end of this process is an increase in disorder, meaning that we were starting, for example, from a portion of space that was filled of gas and another portion of space that was totally empty. If we connect these two portions of space via tube, we will see that the transformation will bring equilibrium, and this is a consequence, or this is measured by the second principle of thermodynamics. Operationally, you measure this by employing the state function entropy and by observing that due to calculation, the entropy of a system tend to spontaneously increase. There are no spontaneous processes whose only result is in the decrease of the overall entropy quantity of a system. Schrödinger noticed that living systems seems to defy this principle. So there's something going on there in which it is not explainable by classical physics, or in this context by classical thermodynamics, because classical thermodynamics told us that the goal function or objective function of any natural transformation process is that of going towards states of high entropies. But if we look at living objects, instead we notice that the living objects tend to display and replicate some kind of structures. I give you the example of plants. For example, plants, one of the fundamental constituents of plants are leaves. Leaves are structure, very well defined structure on their own, so that if we repeat continuously the assemblage or the production of this structure, what we are doing is that we are producing, what it is happening, it is that there is a production of more and more quantity of ordered structures. Sorry. Okay. There are plenty of people that are getting in and out. Okay. So how to cope with, how to put these observations that comes from very simple observation of living object into a framework that is consistent with what we believe it is a fundamental theory of our universe, that is the theory of thermodynamics. So I recall you that the theory of thermodynamics is based on two fundamental principles. The first one is the energy conservation principle, and the second one is the entropy increasing principle. In order to understand what was happening with living things, first of all, Schrödinger posed out two issues. The first one, the first instance, is that of creating order from disorder. So when we focus on living objects, one of the things you may notice of living objects is that these objects are able to assemble ordered structures starting from very disordered sources of materials. This instance, order from disorder, was first demonstrated by some physical experiments in the 60s by Ilya Prigozhin, who has developed the thermodynamics of far from equilibrium processes. And when studying far from equilibrium processes, Prigozhin realized that there were the appearance of some kind of ordered structures that he then called dissipative structures that had the ability of increasing energy dissipation. So energy dissipation, as we will read it further or later, may be seen as energy transformation. I already told you that we go from solar energy in order to create organic matter. This is photosynthesis. But in doing this, a lot more of energy is employed to create a smaller amount of organic matter, smaller amount measured in terms of energy content of organic matter. The difference between the energy employed to produce organic matter and the quantity of organic matter produced is exactly dissipated energy. Energy that somehow has changed its form. For example, from solar, very coherent, highly ordered solar energy, light, into very disordered, highly disordered energy, that is, for example, thermal energy. So, order from disorder appears from due to dissipative processes in which some energy is simply consumed or thrown away, and in doing this process of dissipation, ordered structures are created. These structures I'm talking about structure, dissipative structures appear in the reality. There are some kind of experiments. For example, the Benard cell experiment, in which you have a very thin layer of water, but is bounded, limited in between two metal plates, very small, close metal plates, some millimeters. So we have two metal plates that are displaced of some millimeters. And in this space, in the space between the two metal plates, you fill with some liquid, let's say water. Then if you start heating one of the two plates, you will see that by various energy transfer processes, you will have some energy that will move from the hotter plate to the cooler plate. And first of all, you will have radiation processes. So energy is transferred to processes. And you increase the temperature difference between the two plates, then the transfer mechanism will pass from irradiation, thermal radiation to convective motions. What happens into the Banach cell experiments is that if you increase the difference, the gradient in temperature between the two plates, then at a certain point, at a critical value of the gradient of the difference in temperature, you will have some kind of structures, convecting structures that appears, the Banach cell structures. And so this is a kind of appearance of order. So this is what Prigogine meant by order from disorder. Schrödinger in 1944 also proposed another important issue. It is that of order from order. He said that somehow, given that it is apparent that living systems have the ability of duplicate themselves in a very robust way, Schrödinger has the intuition that somewhere in the living object should be coded or contained the information for building the structure of a living organism. This order from order issues that was proposed by Schrödinger in 1944, it is a surprising anticipation of a discovery of a chromosome or genetic code DNA that happened in the 50s. I recall you that DNA was first discovered some years later in the beginning of the 50s. What happened, what appeared to be relevant to Schrödinger, that was a physicist, in particular, he was a quantum mechanic physicist, so his principal tool was statistical mechanics. Schrödinger realized that the possibility of this kind of changes was linked to the fact that there was a very large number of elements that were collaborating to the various living phenomena. It has to do, for example, with the law of Penrose of inaccuracy that tells us that the inaccuracy of our observations decrease as the square of n with the increase of the number of objects. So it is objects involved into the process. So it is a first indication that there is a kind of interplaying of several objects that make up the evolutive process, so the dynamic process of evolution or of life. It was Schrödinger itself that has the intuition, because he was a physicist, but there should have also been a mechanism of mutation, and the mechanisms of mutation was addressed by Schrödinger was X radiation, what we call hard radiation. X radiation is a high energy radiation that is able to modify in a significant way the behavior of a system. Please note that all these reasonings, all these considerations came half a century after Darwin stating of a theory of evolution. I recall you that in the theory of evolution there are a couple of the key mechanism is the survival of the fittest. This means that we have first a mechanism but of mutation. So mutation of a genetic code generate different organisms, and the different organisms may be successful if they are fit to the surrounding environment. So the idea is that evolution, that is from a physicist point of view, essentially a statistical phenomenon, is somehow governed or constrained by the evolutionary principle, that is the survival of the fittest. So the two mechanisms are mutation, that generate new organisms, and survival of the fittest, or if you want, adaptation to the surrounding environment. Then there is a third mechanism, is that of propagation. The idea is that those mutations that are successful in terms of survival of the fittest, then will be propagated to the offsprings. So there will be the propagation process that is governed by the DNA mechanisms. Okay, this is strikingly impressive, at least for the scientists of the beginning of the 20th century. But one of the first starting point, from which we have already started our discussion, especially when defining ecosystem, is that living objects seems to need borders and membranes. We already said that life is something that happens into kind of protected environment. What it is, a protected environment? A protected environment is an environment bounded by some kind of border. In living system, the border may be identified with membranes. Please note that from the philosophical point of view, this is a very general discussion. Now I told you membranes, because living things border their space through membranes, but the concept, the very general concept, is space delimitation. So the idea that was brought out by the biologist Varela's, what is life? Life is something that happens when some kind of differentiation in the primeval soup of molecules or of elements, of chemical elements, some kind of differentiation happens. For example, some kind of molecules are differentiated so that for some reason that we still do not understand completely, we have some cells that, or some elements that may be able to delimitate the space. In this case, the bluish elements are somehow bounding a portion of the environment, of a portion of space in which there are these reddish elements, but instead are those that we would call living elements or living springs, living seeds, seeds for living objects. The idea of Varela, following the reasoning of Schrödinger, is that some kind of photons, space radiation, I have indicated here with term h nu. Nu is the frequency, h is the Boltzmann constant, so this is the energy transferred or transported by a photon. These photons may change some of the molecules in the primeval soup so that some of these molecules may be employed for constructing some kind of borders and membranes. It is interesting to push forward this reasoning by observing that if we look at the most simple living forms that we may observe today and that we know are present on our planet since at least 3.5 billion years, but are bacteria. Bacteria are structures. For example, the first bacteria are prokaryotes bacteria that are represented by this kind of schema. There is a membrane, some kind of external organisms, like this in this case, the flagella is a kind of Q or a motile organisms, and the prokaryotes has all the genetic content spread into the cellular liquid. Let's see if the so-called cytoplasm. So the cytoplasm is the cellular bacterial, the content of the bacterial cell. As the first step of evolution, different bacteria that were able to do some different biotechnology start to gather together by a mechanism that is called symbiosis, so that more complicated bacteria types appeared, and these are eukaryotes from the Greek word true. Karyotes means nuclei or kernel, nucleus. So the eukaryotes bacterial cells presents a very particular structure, but is the nuclei, and in the nuclei is where we have the DNA genetic code. So, what is life? First of all, life is something that is linked to the building of membranes, border, an inside that is protected against an outside that is chaotic and unprotected. In order to have life come into play, we have to identify and to find a way to maintain the physical chemical condition constant. And how do bacteria do this? They start by doing building membranes, borders, and first of all, in the very simple first prokaryote bacteria, we have that all the material needed for bacteria is simply grinded into the cytoplasm, while by evolution, some of these bacteria started to work together so that we end up with eukaryote cells that are much more sophisticated living organisms in which we still have a membrane, so a border, but inside instead we have several organelles, like for example the mitochondria or the lysosomes or the Golgi apparatus and so on. All these organelles are floating into the cytoplasm, and there is a very particular structure inside the eukaryote, that is the nucleus or the karyotus, and the nucleus is where you will find the genetic code that seems to be the program that is driving the construction of a living organism. Okay? So now we shall switch our focus by asking us, what is intelligence? Why this? Because it is, at least to our very anthropogenic point of view, clear that somehow living organisms seems to be able to manage information. To manage information, in our language, it's something that has to do with intelligence. Intelligere means to link different levels. I recall you that we already said that living systems has the characteristics of being complex systems. So complexity somehow has to do with intelligence. Complexity means embracing different hierarchical level, while intelligence means managing information or transferring information. Managing information management in anthropic speech may be said to be as knowledge. So, up to a certain point, in the beginning of the first part of the 20th century, since the discovery of DNA, it was customary for biologists to think that what it is intelligence or information management had to do with genetic code. So, a parallel question, what is life? Maybe that of what is intelligence. And now we have to go back to the story of the evolution of thinking in life sciences. The typical approach is that of a dogmatic thinking. We started by creationism, that is the oldest way, when the Bible said that the world was created by a divinity. So this is the prototype of a dogmatic thinking. We do not know. We know that something is happening, and we say that this is due to God. Typical dogmatic thinking, and this approach was called the creationism. Please note that creationism dominated life sciences since beginning of life science. Then there was a striking evolution that is, Breeze from the lung, turn off your microphone. Oh. The first improvement from the creationism come from Darwin in the end of the 19th century, beginning of the 20th century. Darwin introduced or described a mechanism he called evolution. So evolutionism is the modern in the 20th century or in the end of 19th century modern way of describing intelligence, complexity, and so on. I told you that Darwin's principle of evolution were based on the idea of mutations, survival of the fittest, so adaptation and transmission of evolutions. This was somehow dramatic because when in the 50s there was the discovery of DNA, we had an enormous spread out of biological science. This that is called molecular biology, in which biologists had the dream or the idea that everything governing concerning the evolutions or concerning the way life system, living system were acting and adapting in our planet, and to do with genetics. You hear this? Maybe this is people that is asking me to join the class. Molecular biology, genetics, spread out very fertile, very huge stream of research, what's called molecular biology. And for half a century, let's say 50 years, biologists were all concentrated in trying to understand how genome worked in governing evolution. But nowadays, we have a more modern synthesis that is called the neo-Darwinism, in which it is not only DNA, but is describing evolution, or that is describing the complexity or the intelligence of living organisms, but there is something more sophisticated, but that to do with the ability of living objects to interact with their surrounding environment. So the question, what is life? We have recasted this question in what it is intelligence, because we said that life is characteristics of complex systems that may be said to be intelligent or knowledge system. And then now we are at the point of asking ourselves if the biological intelligence is completely stored into the genetic code. This was the program of the molecular biologists of the 50s of the last century. Is DNA the biologic intelligence? Now we shall give a look on how DNA is transferred through living organisms. Do you remember what are gonads? Gonads are the primeval cells by which complex or more evoluted living organisms are built upon. The gonads, for example, for mammals are the spermatozoos in males and the ovules in females. So asking ourselves if DNA is biological intelligence may be recast again as a question as if we are thinking by our gonads. So it is our gonads or the cell, the embryonal cells that is transporting intelligence. Of course, we know that it is not so simple, so we have to go back to a very large project, the Genome Project, that was carried from the 80s of last century up to the year 2000, so for 20 years. The Genome Project was driven by Dulbecco scientists, and it was as the topic of the objective of classifying and understanding the meaning of the DNA. The idea that was very appreciated by biologists is that of finding a univocal correspondence between genes and proteins, because we know that genes are containing all the information for coding for proteins. And we know that proteins are the building bricks for living objects. So the question focused by the Genome Project is to understand which gene was coding for which proteins. And we know more or less that living objects, living things displays a large amount of proteins. The number of proteins is maybe roughly estimated into 100,000 proteins. So we are expecting to find that the genetic code contains at least 100,000 genes in order to be able to code for the 100,000 proteins. So during the Genome Project, several genomes were analyzed and codified, and of these genomes, for example, we found that flies have only 25,000 genes, worm had only 10,000 genes, bacteria 5,000 genes. So you see we are at three different hierarchical levels. The first one, that of flies, so that of bacteria, few genes, 5,000. Then we go to worms, that are slightly more elaborated animals, 10,000, and fly, that we may consider quite elaborated living objects, that contain 25,000 genes. Okay, perfect. So now let's see, we that are the most intelligent, how many genes are coded in our genome. Well, the Genome Project ended up with the result that the human genome is made up only of 25,000 genes, like those of a fly. So, by this observation, the genetic determinism does not exist anymore. It is not conceivable, or it is not thinkable, to find the explanation of life only by understanding the working of the genetic code. This was somehow dramatic to the biologist. It was the same level of dramaticity that physicists experienced in the beginning of the 20th century with the introduction of quantum mechanics. So the Genome Project made clear that there was some other mechanisms that were at work in order to govern the evolution of living objects. What these mechanisms may be, let's go back again. Let's go back to Darwin. I recall you that Darwin said that there were three mechanisms: mutations, X-rays or hard cosmic radiation that may induce mutation in the genome. The other important aspect is survival of the fittest, or there is a kind of selection, natural selection mechanism, so that only those mutations that are most able to survive in the environment are those that will be conserved. And the third part was what is called Lamarckism, that was often referred to as inheritance of acquired traits or characteristics. So from the evolutionary point of view, from the evolutionary point of view, we switch from Darwinisms to Lamarckisms, that is Darwinism, that is mutation and selection. Lamarckisms, that is inheritance of acquired traits. So these Lamarckisms tend to describe the fact that successful mutations will be inherited by offsprings. I recall you, Lamarck, you may remember, you have studied Lamarck during your high school classes, and you may remember that Lamarck said that uh, some traits acquired by living organisms may be inherited by offsprings. And the example of criticism of Lamarckian theories said, okay, so you're saying that if I cut the arm of, for example, an organism, if I cut one of his organs, like the arm, then if its offsprings will be come out without the arm? No, this is not simply like that, but instead there are all the three mechanisms at play. So it is not that the mechanism without the arm will be inherited. Instead, if the organism without the animal, it is the fittest, so the most suitable to fit into the actual conditions, then this will be inherited. So it is a modern way of looking at the effect or the way genetic code works, and this is called the epigenetic. Epigenetic is a branch of biology, molecular biology. This branch of molecular biology studies the changes in the phenotype. What is the phenotype? The phenotype is what it is realized by decoding the genotype. Genotype is the information contained to the genome. When there is a mechanism of realization of this information into real living structure, and this mechanism of realization of this genetic information is called epigenetic. Epigenetic is any activity of gene regulation through chemical processes but do not involve changes in the DNA code, but may modify the phenotype of the individual and of the springs. What we are saying in this slide, we are saying that the DNA contains much more information that is needed to build up an organisms. Instead, it will be the interaction with its surrounding environment that will select which of this information will be actually realized by the growth process. Okay? So, what is gene expression or gene expression control? We may say that during morphogenesis, so the changement of the structure, the totipotent stem cells, you may have heard already about stem cells. Stem cells, for example, the gonad cells, the ovuli or the spermatozoos, these are all stem cells. Stem cells are cells that are possibly be transformed into every type of cells in the organism. So these are called by biologists totipotent stem cells. So during morphogenesis, totipotent stem cells become the various pluripotent cell lines of the embryo. So the various pluripotent cell lines may build up, for example, the skin, the eyes, the liver, the brain, the heart, and so on. So all the differentiation of these cells comes from the totipotent stem cells. So from stem cells, this is why stem cells are so important in modern medicine now, because the idea is that of starting from stem cells, and being able to reconstruct every type of tissues on the basis of stem cells. So during morphogenesis, so during the evolution of the embryo, totipotent stem cells differentiates into the pluripotent cell lines of the embryo, which in turn may become fully differentiated cells of the different organs of the final organism. Where does control of this gene expression come from? The answer is that control comes from the external environment. But it is also linked to a kind of growth noise, the so-called growth noise, but has to deal with the stochasticity, with the effect of casual or random mutations. So where we are now, we are saying that life is manifested by the coding of DNA. The coding of DNA, that is the expression, gene expression, is on the hand controlled by the surrounding environment. This means that the same stem cells will end up in different organisms if these cells are living in different environments. So it is a very relevant point in which we have to include the effect of the interaction with the surrounding environment into the organism evolution. So, in order to understand how this control is manifested, we shall go back to our very simple organisms. Let's focus on, for example, on a bacteria. We said that the bacteria, prokaryotes and eukaryotes as well, all have the same characteristics of possessing a membrane, or living organisms, simple living organisms as bacteria, may differentiate the space-time by creating a portion of a space-time that is controlled and constant, and the stability is maintained against the effect of the external perturbations. If we look at the bacteria, then we see that the interface between the living organism, the bacteria, and the surrounding environment is the so-called membrane. So, membrane have a relevant effect in driving evolutions, and in particular, membranes are the interfaces through which external control act onto gene expression. Membranes, cellular membranes are quite complex structure. In this slice, we see that there is a set of, we have lipid layers. This is a polypeptide layer. Between these lipid layers, there are some kind of canals made up of protein that connect the outer part of the membrane into the inner part of the cell. So there is a quite complex interface that regulates in a non-trivial way the exchanges between the external environment and the internal space of a living object. Okay, this is not a biology class, so I'm not going to details of membranes, but it is nevertheless quite interesting to see that the structure of a cell membrane is somehow topologically similar to what we know it is the closest to intelligent behavior object that humankind has been able to build, that is computers. So the cell membrane has a striking similarity with semiconductors. You may recall that semiconductors are layer of silicon. In this layer of silicon, there are some kind of what they call dragons or alterations, but change dramatically the way the object responds to external perturbations. So a semiconductor, it is somehow the most close object to the cellular membrane, you may imagine. So, again, what is intelligence? What is intelligence? What is life? My answer is that intelligence resides in the cognitive model for the interpretation of reality. In terms of bacteria, these are the membranes, the cell membranes. In terms of more complex mechanisms, more complex living organisms, the interface of the cognitive model for the interpretation of reality comes through the senses: eyes, smells, sounds, touch, and so on. So my answer to what is intelligence, my answer is in the cognitive model that the living organism uses for interpretation of reality. In the case of a very simple living forms like bacteria, intelligence resides in the membrane. It's a game of words, membrane. It is probably where intelligence resides in. This way of thinking has some striking consequences, impressive consequences. For example, if you think of the way in which mammals replicate, you may recall that there is an ovulus, the feminine gonad cell. This ovule is then that is feminine. It's then heated by a spermatozoa, that is masculine gonad cell. And at this point, we have the first splitting of a cell. So once the spermatozoa touches the cell membrane or the surface of the ovule cells, along this point of touch, or it is the spermatozoa that is entering the cell, along this point of touch, we have the first subdivision, cell subdivision, so that the ovule cell that has been fecundated by the spermatozoa divides into two cells. When we have the two first cells that are the origins of the new organisms. We are talking about mammals. What it appears striking is that the part of the gonadic cell that is entirely transferred to the two cells that have been subdivided is exactly the cell membrane of the mother. So the cell membrane of the feminine gonadic cell. So if we think that intelligence is in the cell membrane, then automatically we ought to think that intelligence is transferred to offspring by the feminine line, by woman. Okay, we have said that external environment may have an impressive or significant effect on the evolution of, or if you want, the gene expression of DNA cells. Then, sorry, it's on my telecamera, it is reasonable to ask ourselves which kind of signals are to be processed by these cognitive models of external environment. Are they chemical signals or electromagnetic signals? Some consideration: chemical signals, meaning chemical molecules move with the speed of centimeters per second. compared to electromagnetic signals, what the speed of light, that is 10 to the 5 kilometers per second. So you see that chemical signaling is quite sophisticated, but it is at least not as efficient as it is electromagnetic signals in terms of speed of propagation of signals. So the question is, if you were a community of trillions and trillions of cells like larger organisms are, which type of signaling will you apply? Chemical or electromagnetical signaling? Of course, this is just to say to you that you cannot neglect electromagnetic signals when you are trying to understand how gene expression works. It is not only chemical signals, because of course electromagnetic signaling that have a much quicker way of propagation shall or may be expected to have a very relevant effect. This is just to say to you that we still do not exactly know what happens to life or to living organisms when we bound these organisms with electromagnetic signals. So the idea of building electromagnetic, let's say cellular antennas all around our cities is probably not a good idea. We do not have yet clear indication of what this may cause, but we have significant suggestions that electromagnetic signal or electromagnetic waves may interfere in a dramatic way with lives. Okay, I shall stop here today. I'm not going to quantum mechanics and so on. The point is that there is a materialistic prejudice in which we are ought to think that only chemistry was the relevant part for only chemistry was the relevant part for the transmission of signals in living forms. Instead, now we have some indication that electromagnetic fields are also important, so that diseases that may be induced by electromagnetic fields may become an issue of increasing concern. Okay, by now I stop here. If you have any question, I spent some time here and I'm ready to answer to your question. Otherwise,